So this is the species Gordionus wolterstorffii, and this is a horsehair worm, or it belongs to nematomorph. I will play it shortly again so that you see. So it looks a little bit boring, just a brown piece of spaghetti integral which is moving. So it is belonging to the ecdysozoa, to the nematoida, which means they are the sister group to nematodes, and they have their own clade nematomorpha. So what are nematomorphs? Now look at this little video, what you can find on YouTube. It's a little cricket, but they are not crickets, right? This cricket is jumping into a swimming pool. And look what's coming out of this body. So there's a little worm coming out of the body, and sometimes you have 2 or 3 or even more out of this cricket coming, or praying mantis, or so on. So it's obviously kind of a parasite, right? And this is attracting our attention. So this is the life cycle. So it's mainly, as a final host, it's in a meat-eating insects like praying mantis, beetles, and so on. And then they exit the body. They accumulate in the water. We work on a freshwater species. There are also marine ones. And these adults, they copulate, and then they lay millions of eggs. Out of these eggs, a larvae is developing, and this larvae somehow is infecting another species, and therefore then it is kind of eaten by a meat-eating insect, and then this life cycle is closed. But you see all these question marks, and this is our question. So we use the genome to answer these questions, to have material, right? So first, the behavior. How does it change the behavior of the cricket that it suddenly jumps into the water, right? Our hypothesis is that these are neuropeptides likely co-evolved. And so we look in these genomes for similar neuropeptides, which are in the genome of the nematomorph, which also correspond to neuropeptides in the host. Then the development. I'm a developmental biologist, so I'm very interested in the development of them. And then how do they metamorphose to the long worm from this little tiny larvae, and how does this larvae go transfected into the next animal? So I work with Andreas Hejnol, who is the world expert on these animals, and I asked him about each step, and he said, we don't know. We don't know. We don't know. So we don't know much about these, but they are highly interested. So what are we using it for? We look for developmental genes. So do they have the full complement to make the development? You see it on the bottom left. There's a 4D recording of the developmental process. We will also use in situ hybridization on the larva and characterize the larva and try to understand the life cycle in the host. And this, when we compare it with nematodes, because there are also some parasitic nematodes, and even in the clade 1, the first, or clade A, it's called there, no, clade 1, and the sister group to all remaining nematodes, there are some parasites as well. So maybe it was earlier, before the nematodes became non-parasitic. And we look at cell type evolution, and of course we can maybe resolve a little bit of the ecdysozoan phylogeny with this using synteny analysis. So that's it. Thank you for your attention.